many methods for assaying the antibodies. And in the previous segments, in the previous modules, we have discussed a number of them. Now today, in this segment, I will discuss another interesting method to assay the antibody, and that is known as triple antibody sandwich ELISA method. Now this method, triple antibody ELISA method, is also known as indirect ELISA. That's where the indirect ELISA is used to determine the antibody concentration. Now this method is used to identify antibodies in patients' bloods who may be suffering from some kind of infection, and we can determine that there may be a sign of infection in their blood, whether it is the infectious disease or not, it is the virus or whatever. Now with other immunoassays, the layers of reagents that is built up, each dependent on the binding of the previous one. When these layers, it means when these layers are built up, they're dependent upon that with what reagent, the previous reagent which is there, that it is attached or not. Just like a photographic plates, when we develop it and use different reagents to check. Now, this system is used to test patient blood for the presence of hepatitis B virus, for example, HBV, and HBV virus antibodies, the virus which produces those antibodies that can be checked by the indirect ELISA method. Now, HB coating antibody is bound to the wells of microtitre plate. Now, microtitre plate is a plate which is used for ELISA determination. These are the small wells in that plates. And HBV coat proteins that is added to them. So what we do, that in the well of the microtitre plate, HBV coat protein, it is added to them from the blood from where we suspect that there may be HBV, hepatitis V or not. Now the live virus is not used as antigen. Now in this case, the live virus is not used as an antigen, as this would be too dangerous to be used in the laboratory. So you see, the live virus are not taken, rather the virus coat protein, they are used in keeping in view the safety. Now HBV coat protein is made synthetically for the use as antigen in this type of test. So the synthetically, what happened, that HBV coat protein which is present in the HBV virus is prepared synthetically and is used for the test. Why? Because the live virus, remember, cannot be used in this test, rather the synthetic antigen is used. Now what it is done, that after incubation and washing, the patient's serum is added. So what you do, first you took the microtitre plate, added the antivirus, the coat protein there, and now after incubation and washing, the patient's serum, blood serum is taken and it is added, which we suspect that it may contain the antibody. If it contains the antibody, naturally it will react with the antigen if it contains. And anti-human antibody that is the conjugated to an enzyme marker and then added, which will bind to the patient antibody. So what does we do? Then conjugate it to an enzyme marker and then added. Enzyme marker we mean any enzyme which is, as a matter of fact, can become a marker, can become an indicator. It is added and it will bind to the patient's antibodies which are there. Now substrate is then added. Since we have added the enzyme, and it is added to identify samples which are positive. Because there are marker enzymes which will indicate due to some color reaction or some other method that whether that antibody is present or not. The test works well for the diagnosis of HBV infection. Hepatitis B infection जो है, it works for that diagnosis. Now another use of this test is to ensure that the blood donations given for transfusions are free from this virus. So this test is also, because indirect ELISA is also very useful for the blood bank establishment for making the blood which is used or which is donated for further use to check whether it is free from HBV or not. And for that blood, the same procedure is carried out. That is, as we have described earlier, the blood serum is taken, centrifuged, incubated, and can be used for the test. The HBV, the proteins, they are added to the microtitre plates of ELISA. Further, what happens that the marker enzymes are added, and then the patient's blood serum, which is taken there, it takes not much time to make the blood serum and added on that, and if it gives the reaction, then we can determine that whether it is HBV positive or not. And we can be sure that if it is pure, it is free, we can use it in the blood bank for the further transfusion and can help us a lot to make HBV-free donation. So you see that the indirect ELISA method has played a significant role in the diagnosis of HBV and the diagnosis of the HBV tests.